Good day, I am Kathleen Tamayo, and for this report, I will discuss the immunology of human papillomaviruses. We'll start with the description of the microbe, then we'll talk about the diseases it causes, its clinical manifestations, how we diagnose it, and how they are treated. We'll move on to host immune responses and the specific ways by which HPV evades this. And finally, we'll discuss vaccines against HPV and a little bit about how they are developed. HPV belongs to the family Papillomaviridae, and they are so named because some subtypes cause papillomas. It has an icosahedral capsid and has a circular, double-stranded DNA with about 8,000 base pairs. This is a structural representation of HPV-18 and its viral genome. And the genome can be divided into three sections. So the early region encodes 7 viral proteins responsible for virus replication, cell cycle entry, immune modulation, and virus release. So there's E1, E2, E4, E5, E6, 7, and 8. And then the late region, which are highly expressed during the latter stages of infection, it encodes 2 structural proteins, so L1 and L2. And these form the viral capsid. And in this picture, you'll see the top core structure of one L1 monomer. So this is one L1 protein. And this will be relevant when we discuss HPV vaccines later. The LCR, or the long control region, on the other hand, it controls viral replication and transcription and then post-transcriptional control. There are more than 125 types of HPV, and some of those are considered low risk or non-oncogenic, and some are high risk or oncogenic, and some are potentially high-risk HPV types. So low-risk types like the HPV 6 and 11, they are associated with genital warts, so they are non-oncogenic. And on the other hand, the high-risk types like HPV 16 and 18, these are associated with genital cancers. In fact, these two are the most common high-risk genotypes because they cause approximately 70% of all cervical cancers. HPVs are not enveloped and they exclusively infect humans' epithelial cells, specifically the basal keratinocytes. And they can multiply only in the differentiating stratified squamous epithelium, meaning there can be no systemic infection, right? HPV is transmitted through intimate skin-to-skin contact with an infected person, and transmission is most common during vaginal, penile, anal, or oral sex. Auto-inoculation from one body site, like the hands, to another can also occur. Sometimes vertical transmission from an infected mother to her infant can happen and can result in a condition. It's called the juvenile onset recurrent respiratory papillomatosis. And then finally, anal or genital warts may also be transmitted during birth. Now I would like to briefly talk about how HPV replicates. So upon transmission, the virus is able to enter the basal keratinocytes through tiny breaks in the skin. And once HPV reaches the extracellular matrix, some extracellular proteases, they trigger conformational changes in the virus capsid just to enable its uptake via micropinocytosis. And so the virus travels along the endocytic pathway. So like it travels in a retrograde manner until it reaches the nucleus. And then the genome is then released into the nucleus for transcription, translation, and then in benign lesions, the viral DNA remains located extrachromosomally, meaning it is not integrated into the host chromosome. But for malignant lesions, the HPV DNA is integrated into the host chromosome. Right? And so now going back to this figure. Now the basal cell layer undergoes cell division, and then two daughter cells are produced, and then one of those infected cells gets pushed up into the epithelial surface and then it undergoes maturation, differentiation as it moves until it reaches the upper layer of the epithelium. And then the infectious variants are going to be shed on the surface, right? During normal shedding of the skin or like desquamation. So in low risk HPV, those extra cells cause the warts, which is a potential symptom of HPV. In daughter cells infected with high risk HPV types, we mentioned earlier that the HPV DNA is integrated into the host genome, resulting to increased expression of viral proteins like E6 and E7, we mentioned earlier. And E6 and 7 are oncoproteins, meaning they mediate the development of cancer, some type of cancer, specifically cervical cancer. And these interact with cellular proteins from the host to induce massive proliferation of cells and essentially immortalization. And because of this non-scheduled replication, no, you see an accumulation of cells leading to the disruption of the normal appearance of the epithelial tissue. And the degree of this disorganization is used to describe the lesion. And when these abnormal cells breach the basal cell membrane, that's when invasive cervical cancer happens. Individual types of HPV are associated with a specific clinical manifestation, and the disease they cause can be categorized according to the tissue they infect. So it's either cutaneous HPV or those that infect the skin, or mucosal HPV, which means these infect the mucosal membranes. There are several types of HPV infections in internal skin, and the wart types vary depending on the affected body part. So there are common warts of the hands and face, there are plain warts located in the face, and then plantar warts located in the feet. And they have different causal organisms. And they can present as benign lesions with hypertrophy of all layers of the dermis, meaning there's enlargement of tissue because of the increase in the size of the cells. And this can result in acanthosis or thickening, papillomatosis, or folding, and then hyperkeratosis or increase in the horny layer. This can be diagnosed via visual inspection, and sometimes a biopsy may be taken to test for HPV. For cutaneous HPV infections, you can apply topical medicine so that blisters can form under the wart, and this will cut off its blood supply. You can also apply salicylic acid solutions, which will dissolve the warts one layer at a time. And then cryotherapy, this includes the application of liquid nitrogen to freeze the wart, and then it will induce blister formation, which will cause the peeling off of the wart. And laser treatment, this is where you use heat and destroy tiny blood vessels inside the wart to cut off its blood supply. There also are several types of HPV in mucosal and anogenital skin. So these may include genital warts in male and female, and this is technically called the condyloma acuminatum. All right. Under these type are cervical cancers and anal cancers. And the causative organism for genital warts is the we talked about earlier HPV 6 and 11. For cancers, mainly HPV 16 and 18, and some other types then for anal cancer. They can present, these genital warts can present on the moist tissues of the anus or genital area, although they may also occasionally develop in the mouth or throat after oral sexual contact with an infected partner, and they may be of different architecture. They can manifest individually or by group, and they may start as small but grow several inches in diameter. They may present in different colors. You can diagnose genital warts by history and physical examination of the lesions, and you can also perform confirmatory biopsy, but it's generally unnecessary, or you'll do this if there is uncertainty about the diagnosis or if the patient is immunocompromised. And to treat genital warts, there are destructive and surgical methods, or you can use topical agents. So, uh, destructive and surgical methods are more effective on keratinized warts, especially if they are larger. And these procedures have varying pros and cons depending on whether or not they leave a scar, how much they cost, and if they require sophisticated equipment and training. And then trichloroacetic acid, it's a chemically destructive acid that burns and erodes the skin and mucosa. And then for topical agents, they have different modes of action. Like for this agent, it will bind to cellular microtubules, um, and and then it can inhibit mitotic cell division and induce the necrosis of warts. And then this topical agent, it's an immune enhancer. It will induce the secretion of cytokines that reduce the HPV DNA viral load. Sorry. For cervical cancer, it can manifest as postcoital spotting, intramenstrual cycle, bleeding, foul smelling or persistent yellow discharge, and or pelvic or sacral pain. And then you can diagnose it via pap staining of smears. And in infected cells, you are going to see coilocytes, which is a type of epithelial cell that develops following HPV infection. And then you can also do HPV testing by PCR. And the choice of cervical cancer treatment may include cervical removal, cryotherapy, laser therapy, radiotherapy, and hormone chemotherapy, depending on factors like stage of disease, type of tumor, and degree of tumor differentiation. And finally, for anal cancer, it may manifest as benign lesions that arise in the anal canal, but also a mass or a growth may appear in the later stages. And then there are lots of other manifestations. And you can diagnose it by anal pap smears, also through physical examination and high resolution anoscopy, wherein you examine the anus for abnormal cells that have a high likelihood of turning into cancer. And then you can also do a combination of these treatments to eliminate or to treat anal cancer. Diseases under oral HPV include oral papillomas and HPV associated with oral cancers. And these are the causal organisms of these diseases. They may present as, or sorry, Oral papillomas may present as common warts, which can be spread from the hands to the lips by finger sucking or nail biting. And then there are oral lesions, there are oral squamous papilloma, oral condyloma acuminata, which is characterized by little whitish or pinkish nodules, and all of this proliferate over the tongue, lips, palate, and floor of the mouth. Meanwhile, HPV-associated oral cancers may manifest as long-lasting sore throat and earaches, hoarseness, swollen lymph nodes. And both of these diseases you can diagnose via screening or examination by the doctor and to inspect the throat or the larynx or the base of the tongue. You can do laryngoscopy and pharyngoscopy. And then finally, biopsy can be done. And then you do microscopic examination and testing for HPV DNA. Corticosteroids may reduce the symptoms, but it will not cure the condition. And then you can also treat with calcineurin inhibitors and topical retinoids. And then your usual combination of HPV treatment. Different innate immune responses allows the host to fight off this viral pathogen, and some of those are physical barriers. So recall that the native host cell of HPV are the basal keratinocytes, and this preference or tropism actually plays to our advantage, right? Because HPV first needs to translocate across skin and mucous membranes. But these mucous membranes is a physical barrier to virus infection because they can secrete viscous protective fluid and antimicrobial peptides. And once HPV reaches the extracellular matrix, we discussed earlier the retrograde transport of the virus. In this process, majority of the virus particles are degraded and eliminated by host autophagy, which is a self-digestive pathway involving the lysosomes, which is where the virus travels along, right? Basal keratinocytes are also equipped to sense pathogens because they express PRRs or pathogen recognition receptors to sense PAMPs. And some of these are nod-like receptors. There's also a high expression of TLR2, 3, 7, 8, and 9. And the high expression of these correlate with the clearance of initial HPV infection. And one of the downstream effects of PRRs is the production of interferon alpha and beta, and specifically, this interferon beta, it hinders or it prevents HPV entry, and it promotes the clearance of latent HPV episomes in case of persistent infection cells. So we talked earlier about certain types of HPV wherein the genetic material is extrachromosomally located. And so interferon beta can clear those extrachromosomally existing HPV genomes. And then innate immune cells are also involved in early host responses. HPV infection can recruit macrophages, dendritic cells, Langerhans cells, natural killer cells into the HPV-infected microenvironment. So these antigen-presenting cells, they capture HPV pathogens and then digest them into peptides. Then they are translocated into the lymph nodes where HPV peptides are exposed on the cell surface via MHC class 2. And then the activated immune cells also release inflammatory cytokines like interleukin 1, IL-6, tumor necrosis factor alpha, and then IL-12, which will induce local inflammation and they can function as danger signals as well. A specific type of dendritic cells, like the plasmacytoid dendritic cells, they respond to the presence of HPV 16, and these can secrete various cytokines like interferon alpha, IL-6, tumor necrosis factor alpha, and IL-8. And then in increased HPV infection and other HPV associated cancer, researchers observed that in the incidence of these diseases has been observed in individuals with various natural killer cell deficiencies. So you would infer that NK cells are part of the body's first line of defense against this viral infection. We talked earlier about how the HPV cycle is intraepithelial, so that's how it's called. The life cycle is intraepithelial. And we discussed also how virions are produced only from the upper layers of skin cell after they are shed, and so away from the circulating immune cells. So this means there can be no cell lysis or viremia, or there can be no virus in the blood, and this limits the exposure of HPV to immune responses, to systemic immune responses. But there are still adaptive immune responses from the host, which we're going to talk about. The signal from APCs that we talked about earlier, this triggers the differentiation of naive CD4 plus cells into type 1 helper T cells and type 2 helper T cells, and they mediate the adaptive immune response during infection. So Th1 cells induce cytotoxic T lymphocytes to kill various infected cells, and they release IL-2, IL-12, and interferon gamma. On the other hand, Th2 will stimulate the humoral immunity, so they stimulate B cells to produce antibodies. A naive CD8 plus T lymphocyte will recognize the MHC class 1 antigen, no, via cross-presentation of the same dendritic cell that presents the MHC class 2 restricted antigen. And so the CD8 plus T cell will differentiate into a cytotoxic T lymphocyte, and then through these cytokines released by Th1, the cytotoxic T lymphocyte will be activated and it becomes an effector T cell that can kill cancer cells expressing HPV pathogens. The lesions caused by low-risk HPVs will eventually go into remission or resolve on its own even without treatment, and this is the result of the cell-mediated immune response. Meanwhile, for humoral immunity, the B cells that was stimulated by Th2 will produce the antibodies against the specific HPV type, and specifically for the L1 major capsid protein, and this will prevent the spread of infection through virus neutralization. So you can see HPV infections still trigger both a humoral and a cellular response in the host immune system, but the antibody levels from these natural infection are usually too low or to guarantee a protective effect, and this suggests that HPV is able to evade the host antibody response. So it is worthwhile to look into the immune evasion mechanisms of HPV and how it contributes to the persistence of the disease. So these are some of the immune evasion mechanisms of HPV and the keratinocytes. So they can suppress TLR9, type 1 interferons, and CXCL14 transcription. And as a result, the host cell will have problems sensing specific danger signals from suppressing TLR9, and it cannot produce the CXCL14 chemokine. Then there will be a proviral state because if you recall, type 1 interferons promote an antiviral state. For example, by inhibiting viral replication. So, by suppressing this, there will be a pro-viral state. They also block pathogen recognition receptors, signal transduction, and prevent the nuclear translocation of this, which means the transcriptional induction of pro-inflammatory cytokines, chemokines, and additional inflammatory mediators will be prevented. And lastly, HPV can suppress antigen presentation by MHC class 1 and CD1D because they interact with the MHC1 promoter site and they can block the transport of MHC and CD1D proteins into the cell surface. And this is how they can avoid detection of cytotoxic T lymphocytes. And all of these are mediated by the oncoproteins we talked about earlier, the E5, 6, and 7, and then E5 also. They can also impair the function of immune cells. For example, they can downregulate certain chemoattractants and cell adhesion molecules in HPV infected cells. So they can downregulate specifically CCL20 and E-cadherin. And if these are downregulated, it will reduce the amount of Langerhans cells that can infiltrate these HPV infected cells. They can also upregulate tumor derived factors and they can downregulate type 1 interferons in HPV associated tumors. Again, these interferons promote an antiviral state. And so if they are downregulated, it can suppress natural killer cell activation and its killing capacity against tumor cells. And then they can also cause the accumulation of these cells, tumor-associated macrophages and myeloid-derived suppressor cells. And what are their significance? No? These myeloid-derived suppressor cells are actually newly identified immature myeloid cells that can suppress immune response. So if you accumulate these kinds of cells, the immune responses will be suppressed. And actually, you can see in this, you can see in this picture the elaboration of interleukin 10, interleukin 10, which we know from our discussion, it limits host immune response to pathogens so as to prevent damage to the host. And then finally, HPV can promote the accumulation of regulatory T cells and a shift from a Th1 response toward a Th2 response in local microenvironment. And both of these actions, including the shift from Th1 to Th2 responses, are mechanisms to avoid a prolonged inflammatory responses. And this is why HPV persists for so long that it gets the chance to develop into cancer. And this is also the reason why consistent checkups every 3 to 5 years is suggested. And then again, the sense of dormancy in the HPV life cycle that we talked about, its intraepithelial life cycle, the lack of viremia, the lack of cell lysis or cell death, the infected cells expressing abundant viral proteins being shed away from circulating immune cells. These are all immune evasion mechanisms. At present, there are three HPV vaccines licensed for use. There's a bivalent, quadrivalent, and a nine-valent vaccine, which offers protection from at most 9 different types of HPV, including HPV 16, 18, 6, and 11. And these are recombinant vaccines produced through recombinant DNA technology. And if you remember from our lecture, in this type of vaccine, you get the DNA encoding an antigen. So you get that, and it should be responsible for stimulating an immune response. And then you insert it into a bacterian or a mammalian host cell, and then the antigens will be expressed in these cells. Then you can get it and purify it and develop it into a vaccine. So let's apply that knowledge in the context of the HPV vaccine. So it was observed that when L1 and L2 proteins are expressed in vitro, they will self-assemble into a structure just like the viral capsid, no? And it is called the virus-like particle or VLP. And then the VLP can induce the immune response, which is similar to a live virus, but it will not produce infection in the recipient because it lacks the viral nucleic acid. So, for this vaccine, you have VLPs for a specific, so per vaccine, no, you have VLPs for a specific type of HPV. And in preparing the vaccine, the L1 gene was inserted into a plasmid and was introduced into a host cell. And then different vaccines made use of different host cells, as you can see in this table. And then upon translation, capsid proteins were produced, which assembled into empty viral capsids or VLPs, just like what happened in vitro. And these were harvested and purified and then developed into a vaccine. And it was coupled with different types of adjuvants to enhance its immunogenicity to weak antigens. This is the end of my report. I hope you learned a lot. Thank you very much for your attention.